Welcome to the Peak Studio DDA workflow video tutorial. In this video, I will explain how to set up a DDA analysis and explain the result structure. To begin, click on New Project. Name your project and change the saved project location if desired. Click on Add and locate your raw data. Peaks is a vendor-neutral software, so there is no need to convert the raw data to another format prior to loading into Peaks. Click this top button to add each file to one sample. This option is for fractionated data. The second button will add each file as its own sample. The third button will allow you to add additional fractions to an already existing sample. And the fourth button allows you to create new samples using regex. With regex, you can auto-name your samples based on the existing sample name. Now, choose your enzyme, instrument type, fragmentation method, and acquisition method from each drop-down menu. If you do not see your desired enzyme or instrument type, you can configure custom ones under the Configurations tab. Click on a sample and then choose Copy to Whole Project to copy the chosen settings to all samples. You now have the option to choose Submit, which will load the samples without running any analysis. You will then be able to go back later and set up a workflow. For this demo, however, we will continue with the rest of the setup. Click on Next to continue. The workflows in Peaks Studio have been reorganized and simplified. Workflows with common steps have been combined into one, with the option to finish the analysis at the desired step. The two workflow choices are Proteome, which includes the traditional de novo assisted database search and reports at the protein level, or Peptidome, which incorporates our deep Novo algorithm and reports at the peptide level. For this tutorial, we will focus on the Proteome workflow. Next are the data refine options. As part of data refinement, Peaks will perform centroiding, deisotoping, baseline correction, and charge deconvolution on MS2 spectra. If you have already done this yourself, you can choose pre-processed data, and Peaks will skip these refinement steps. If you are unsure, then keep this unchecked. Mass correction will correct the precursor M over Z to the monoisotopic M over Z of the feature detected by Peaks. This feature is useful in scenarios where the monoisotopic ion was not selected as the precursor by the instrument. Associate feature with chimera allows Peaks to detect when an MS2 spectrum contains fragment ions from more than one overlapping feature. We recommend leaving both mass correction and associate feature with chimera on. There is also the option to filter features by M over Z, retention time, charge, and abundance if you wish to exclude a part of the data from the search. On this page, you can set up the de novo sequencing parameters, which are now separate from the database search parameters. Start by choosing either the de novo or the deep Novo algorithm. If you choose de novo, you will be able to run either a standalone de novo sequencing analysis or a de novo assisted database search. If you choose deep Novo, then you will be able to run a standalone sequencing workflow with FDR control. However, you will not be able to continue on to a database search if this algorithm is selected. For a combined deep Novo and database search analysis, select the Peptidome workflow instead. Choose your precursor and fragment error tolerances. You can also select an enzyme here to override what you selected in the project creation step. Be sure to choose specified by each sample if you have multi-enzyme digests. Set your fixed and variable PTMs. Peaks has a comprehensive list of built-in custom modifications, but custom PTMs can be configured if desired. Set the maximum variable PTMs that you wish to allow per peptide. If you want to run de novo sequencing only and no database search, choose Report and then go on to submit the analysis. If you want to move on and add a database search, click on Next. On this page, you can set up the de novo sequencing parameters, which are now separate from the database search parameters. Start by choosing either the de novo or the deep novo algorithm. If you choose de novo, you will be able to run either a standalone de novo sequencing analysis or a de novo assisted database search. If you choose deep novo, then you will be able to run a standalone sequencing workflow with FDR control. However, you will not be able to continue on to a database search if this algorithm is selected. For a combined deep novo and database search analysis, select the Peptidome workflow instead. Choose your precursor and fragment error tolerances. You can also select an enzyme here to override what you selected in the project creation step. Be sure to choose Specified by each sample if you have multi-enzyme digests. Set your fixed and variable PTMs. Peaks has a comprehensive list of built-in custom modifications, but custom PTMs can be configured as well if desired. Set the maximum variable PTMs you wish to allow per peptide. If you want to run de novo sequencing only and no database search, choose Report and then submit the analysis. If you want to move on and add a database search, click on Next. This page is where you set your database search parameters. Any settings you chose in the de novo step will be auto-filled here too, so you don't need to re-input everything. However, you can choose different settings if you wish. Peaks does an in silico digest of your provided database, which comes with some additional options, such as setting the number of missed cleavages allowed. Additionally, setting the digest mode to specific means that the digestion rules are obeyed on both ends of the peptide. Semi-specific allows for one end of the peptide to disobey the set digestion rules. If none is chosen as the enzyme, then the digest mode options change. Unspecific means that the database will be digested with no specificity. No digestion means that the database will not be in silico digested at all. This option should only be chosen if your database already consists of peptide sequences and not protein sequences. The target database can either be configured ahead of time or here in the project wizard. Click on New to configure a database. Choose the FASTA format, name the database, and browse for your FASTA file. Click on Validate to double-check that Peaks can parse the headers correctly and check for invalid content. This step is optional. Click on Add Update to configure the database. If you are using a multi-species database from Uniprot, the taxonomy can also be filtered here. Pay attention to the number of sequences listed here and make sure that it matches the expected number. A contaminant database can also be added. The contaminant database sequences will be concatenated to your target database and they will be searched together in a single round search. A limit on peptide lengths to be searched can also be set here to a maximum of 65 amino acids. If the Deep Learning Boost box is checked, then peaks will use predicted retention time, ion mobility, and fragmentation patterns to rescore the peptide spectrum matches. This can be unchecked if desired. Selecting Peaks PTM will allow more PTMs in addition to the ones specified to be searched. Choose either all built-in modifications or select a more focused list. You can also have peaks find potential signature ions for PTMs, which can then be incorporated later into the PTM information if you would like. By default, only spectra with a de novo average local confidence score of 15% or greater will be included in the Peaks PTM search, but this can be changed. Select the Spider checkbox to search for additional mutations and sequence variants. If you select Peaks PTM and Spider, then peptides found by these search algorithms will be included in any subsequent quantification steps. We can now go directly to the report page from here, or we can click Next to have the option to add a quantification or quality control step. Let's click Next. Quantification will be covered in another video, so let's leave it unchecked for now to skip it. Quality control is an optional step that will provide statistics about your data based on different attributes that can be selected. It will let you know whether each attribute falls within the set acceptance tolerance from the control. This works best for assessing reproducibility and can be helpful for figuring out why a sample may be deviating from others. The results from QC do not impact your search results at all. It is only meant to provide additional information. Standard peptides can also be indicated here. The final step is the report filter page. You can filter your results based on a specific -10 log P score cutoff or a false discovery rate at the peptide spectrum match level or the peptide level. For amino acid level confidence, the direct supporting ions must be at least 2% of the base peak by default. This can be changed here. Filters can also be set at the protein level, as well as the number of unique peptides required for a protein to be reported. A minimum ALC score cutoff can be set for de novo sequenced peptides that were not found in the database. De novo tag sharing allows for de novo only peptides with at least the specified number of amino acids common to a database sequence to be shown in the protein coverage view. Checking de novo only fully matched will only include sequences that fully match the database sequence. The workflow parameters can be saved, so you will not have to re-input any if you wish to analyze other data with these same parameters. You can give your analysis a name here, which can be changed later. Batch submission will create a separate result with this set of parameters for each sample. Batch submission cannot be checked if QC is also selected. And you can choose to export your results here. This will export all of the possible result files, which may increase analysis time. You can also pick and choose what you want to export later after the analysis is complete. Click on Submit to start the analysis. Now let's take a look at a completed demo project. The result layout is quite comprehensive, and so we won't be covering everything in detail in this video. Rather, this will be a general overview of the result structure. For detailed explanations of each figure and the contents of each of the result tabs, please see your user manual found within your Peak Studio installation folder. In the upper left window, you will see your project name. Underneath will be any analyses you have run, along with the loaded data. Expand a data file to view the raw data. Peaks will give you an MS1 focused view, an MS2 focused view, and an LCMS map. Expand an analysis to see the refined data and result nodes. Like with the raw data, you can expand the Data Refine node and view the refined data. The data view will include the preprocessing information as well as result information. The LC-MS map can show detected features, MS-MS spectra, de novo peptides, and peptides identified by the database. In a DDA database search analysis, you will see a de novo and a database node. If quantification or QC was run, then those would also appear as separate nodes. Double-click on a result node to view it. Since the first step of the de novo assisted database search is de novo sequencing on all MS2 spectra, these de novo sequencing results can be viewed on their own. The de novo result is reported prior to the database search step and thus will not contain any information from the database search. In this result, you'll see a summary tab with result statistics, a de novo tab with the de novo sequencing results on each scan, and a feature tab where sequence peptides are grouped by feature ID. The DB search result node will contain the peaks PTM and spider results in addition to the database search results. The summary view provides statistics about the results. The proteins view shows the coverage of each protein identified with a list of supporting peptides and de novo only tags. The Peptides tab lists each identified peptide with scoring information. The corresponding MS2 spectrum is shown below in a mirror plot against the predicted spectrum. This can be changed to an annotated spectrum. By default, the highest scoring spectrum is shown, but other spectra that support this peptide ID can be selected with the breadcrumb feature. The Modified Peptides tab shows modified unmodified pairs in a mirror plot with color-coded PTM confidence levels. Similarly, the Mutated Peptides tab shows the peptides with sequence variants against their corresponding wild type. The de novo only tab lists the peptides that were sequenced at the de novo sequencing step with a confidence that passed the user set threshold but were not identified in the database search step. Finally, the feature tab shows the identifications from all steps, grouped by feature. Peptides identified in the database search will have their sequence listed under Identification Peptide, while peptides only identified by de novo sequencing will have their sequence shown in the de novo peptide column. Click on the Export node for all of the export options. These are grouped by the initial de novo sequencing step and the database search step, which again contains all peaks PTM and spider results as well. Check the options that you wish to export, choose a location, and then click Export. We hope you found this Peak Studio DDA tutorial helpful. Please visit our website at bioinfor.com for more information, answers to frequently asked questions, and other tutorials. Thank you for listening.